All right, welcome to chapter 11 and chapter 12. So for today, we are covering cell communication and the cell cycle. So, cell communication. What do y'all know about cell communication?
Literally nothing, because I don't understand shit that she teaches, because when she teaches, she says, I don't
Luis, calm down, calm down. This is going to be posted on YouTube.
Hey, this is my bad. Yeah, life.
Okay.
Cells send messages in order to get a response
Yes,
to stimuli?
yes, that's the main point of cell communication. So with cell communication, cells need to talk to each other in order to do things, especially if you're a multicellular organism with certain special functions. Like if you have, too much blood sugar, right? Your body needs to send a signal in order for you to reduce the amount of sugar in your blood so that, bad things don't start happening. And so when we talk about cell signals, the main things we need to know are the parts of it, right? We have a cell. That's going to be how we represent cells for today. I might draw them in blocks, who knows? Each cell, in order to receive a message, must have a receptor, and draw it like that. And for a message to be sent or received, the cell must receive a ligand. Oh, that's terrible. Hang on. That's even worse. Give me 1 second. There we go. Okay, that's close enough. So a ligand is something that binds to the receptor, like a substrate binds to an enzyme, and then that cell receives the message because this binds here. The receptors are also specific because they're like enzymes, right? They're kind of like enzymes. When they bind to the ligand, it has to be that specific ligand in order for a series of things to happen in order for the signal to be sent. Does that make sense?
Yes.
Yes? Okay. So first thing we need to cover is, well, distance, right? So the main thing about distance is cells can communicate across many distances. Cells can communicate with themselves. Cells can communicate with its neighbors. Cells can communicate by connecting to each other, and cells can communicate between very, very long distances. Let's start with cells communicating with themselves. We call this autocrine. Auto meaning self, right? So I have my cell, and certain cells will produce certain ligands, right? That's how signals get across. Something needs to produce the ligand, right? So when a cell decides to communicate with itself, it's going to produce the ligand, it's going to send the ligand out, and that ligand is going to find a receptor. In this case, it is communicating with itself. So the ligand is just going to meet with the receptor on the cell, and the message is going to trigger. Now, why does it need to go on the outside? Because the receptor is on the outside. That part makes sense?
Yes.
This is autocrine communication. In paracrine communication, a cell sends a signal to its neighbor. In this case, the cell on the left is going to be the one sending our ligand out. So it's going to produce a ligand. It's going to send it out. This ligand is going to make it all the way over here. And then its neighboring cell will have a receptor that matches. And the ligand will meet. This is paracrine. There's some distance, but it's not that far. Does that make sense? Yes, yes, yes.
Wait, autocrine and paracrine are just the name of the process, or what?
It's the name. It refers to like the distance, right? Autocrine.
Oh, just
communicating
the distance.
with
So it's the same process, but it's just the distance has different names?
Yeah, pretty much.
Okay.
This is just how to send a message. So that's autocrine and paracrine. Everybody understand those two? Yes, yes, yes.
Yes.
All right. Let's talk about juxtacrine. Now, juxtacrine, well, the cell isn't communicating with itself, it's still communicating with the neighbor, but this time there's a channel. The cells have formed a channel, and one ligand from in here, produced by our left cell, is simply going to follow through that channel and go into the right cell, where there's probably a receptor on the inside, right? Does that make sense?
But didn't you say like the receptor's always on the
Wait,
outside?
can you repeat that?
So for juxtacrine, one cell which produces a ligand will form a channel with another cell. In this case, we see the channel here. And the ligand will go from the left cell, which produced it, into the right cell. Now, to answer Alex's question on why can it do that, there are receptors that are inside the cell. They're not always on the outside. There are interior receptors. All right, everybody understand that?
Yes.
Yes, okay, cool. Next thing, let's talk about endocrine. Oh, whoops. Endocrine. Well, I have a cell here, and it's going to produce my ligand. But it wants to send its message to a cell over here. And coincidentally, there happens to be a very neat highway for it to use. What is this?
The endocrine system?
Yes. Or, in this case, I drew it in red. It can be the endocrine system, or it can just be a blood vessel or a vein. Right? Because that's where things go. So for endocrine, we're talking about big, long distances, right? Hormones produced in your brain, in this case, the ligand is the hormone. Hormones produced in your brain need to go into other locations. Well, they're going to use the endocrine system or the vein or your blood to get there. And so this cell is going to send its ligand into this blood vein. It's going to travel along this. It's going to exit and attach to that cell. I'm not going to draw the receptor or anything because I'm pretty sure y'all can understand that. That makes sense? Fairly easy. Right? All of that makes sense?
Yes.
Yes, okay, good. So now we've talked about how signals get to other cells. Now let's talk about receptors, right? Now receptors are very important. So let's magnify this. This is a cell membrane. I'm going to draw a receptor there, but just know this entire border is a cell membrane. Let's see, I'm going to do that. And then let's draw a ligand. So the ligand is now attached here. It's kind of ugly, but you get the point, right? It's attached. It's supposed to perfectly match. And now we need to go through the 3 steps of cell signaling, right? Because you don't just instantly get a reaction whenever you make contact with the ligand, right? Because that can cause certain problems. What if your body's producing too much of a ligand? Well, now you're consuming, so now that reaction is happening too much, and your body's going to go haywire. So there's a process for receiving signals. Step 1. Is reception. Reception is the ligand connecting to the receptor, right? Now, when the ligand hits the receptor and it matches perfectly, I'm going to draw this green. A little message is going to trigger here, right? Now we hit step 2. Well, this message doesn't just stay in the receptor itself. It needs to travel through a series of things in order to trigger a response. So step 2, we're going to call transduction. Transduction is a series of processes that eventually trigger your response. What it does is, well, there's a protein, or a couple proteins usually, and this protein will receive the signal, usually by making physical contact, but in this case I didn't draw it close enough. But usually it will make physical contact with the receptor. That signal will travel into here in a physical form, usually changing the shape of the protein, right? The physical form of a signal is the change in the shape of the protein. I am emphasizing my words so you write it down. This protein, which now has the signal, aka its shape has changed, is then going to make contact with this other protein we have here. And it is going to send the physical form of the signal into this protein. So now it's changed. And usually there would be more proteins in this process, but that's going to keep happening until it triggers something that will leave you with step 3, the response. And the response is specific to certain things. So let's say, for example, I'm going to draw this in dark blue. That's my nucleus. For example, if that's my nucleus, right? And the physical form of this message is trying to tell the nucleus to start replicating DNA, right? For a certain process, or sorry, reading DNA so that it can make more of a specific protein, right? So this protein is going to, well, send its physical signal to a protein inside the nucleus. And once that protein receives the signal, it knows, aha, time to scan some DNA. And then that protein will perform its function. Does all of this make sense? Yeah, just got a chat message. Yee. All right, everybody else clear on this, hopefully? That is all the steps of cell signaling. The ligand needs to be transported through either autocrine, paracrine, juxtacrine, or an endocrine process, and then it needs to be received in step 1, transduced through a series of proteins, and then a response can trigger. So that's all your steps, but we are not done with chapter 11 just yet. One more thing, we need to talk about the 4 major receptors. So your 4 major receptors, the first of which is the G protein. Let me redraw that membrane once again. This time we're going to have 2 unique proteins. Good old Pac-Man shaped protein. And then I'm going to do this, it does that, and it does that. And there is this thing here. So in this model, I have drawn you, well, almost everything. So to explain the unique part of a G protein, it's, well, the G protein, right? So for a G protein receptor, the process is going to happen like this, right? Rather than following the 3 steps, it is going to do something similar, right? What's going to happen is the ligand is going to make contact with the receptor. The receptor is then going to receive a message. that message is then going to trigger the activation of this G protein that I have under it and is connected to it. So in order for this G protein to properly activate, a new compound called GTP, similar to ATP, but with, G base instead, is then going to replace a different compound called, or GDP. GDP, when it is connected to the G protein, signifies that that G protein is inactive. Does that make sense?
Not really, no.
Okay, let me restart then.
It's basically like a switch, pretty much, right?
Yes, it's a switch. So GDP is going to be here. When the signal is received by the receptor, a GTP is going to make contact with the G protein, and it is going to move the, hang on, where's the cursor button? I know it's around here. Okay. It's going to take this GDP, is going to move, is going to move it out, and instead, our GTP is going to move in. And when the GTP makes contact with that G protein, wait a second, I gotta move all this, if it'll comply with me. And now that that GTP is connected to that G protein, it's going to tell the G protein to move. And the G protein is going to move right on over to this slot, which perfectly fits it. Y'all understand up to that part, right?
Why would it want to do this?
Why would it want to do this? I will explain once I'm done with the process.
Okay.
So now your GTP is connected to your G protein, and it's telling the protein to move. And when the protein moves, it is going to fit perfectly into that new spot that we've made for it. Let me just erase all this. That GT, or the G protein, is going to fit right in here. I'm just going to label it with a G, and there's a GTP connected to it. Just going to write GTP. And when it makes contact with this, it is going to activate this protein, which is going to then open a channel. And when the channel is open, things can start going in and out. So now things can go in and out. Say, for instance, oh, whoops, that's terrible. Hang on. Glucose molecule, right? When the G protein makes contact here, the channel opens and the cell says, oh, got to take this bad boy in, right?
I get it now.
Yeah, that's the point. But you can't just leave this channel open forever, right? So in order to deactivate the G protein, right, I need to connect it to a GTP. GTP. It needs to connect to a GDP. Sorry. So in order to replace the GTP, I must hydrolyze the GTP, aka break it apart, right, through hydrolysis. And when you break a GTP apart, what you get is a
GDP.
GDP, that's correct, and a phosphorus, which happens to fall off. And when the GTP turns into GDP and is now connected to the G molecule, the G protein will then return to its original position with a GDP connected. And when you do that, we are then back to the beginning with a GDP here. And at this point, the ligand is gone. Ta-da, now we're back at the beginning. Oh, that makes sense?
Yes.
Now, you don't only perform this process for glucose, you perform it for other compounds. Notably, though, you do not perform this for ions. So you don't perform this for non-organic things like salt. So, everybody understand G proteins? Did I lose some people?
Yes.
No, like, did people leave? Oh, that sucks. All right. Everybody ready for the next one?
Yes.
All right. Next one is tyrosine kinase. Tyrosine kinase. Well, tyrosine kinase actually looks a little something like this. Right. And there's your cell membrane. And I accidentally didn't leave a spot for the receptor, so it's just going to be a blob this time. So when the receptor connects to that and activates tyrosine kinase, what tyrosine kinase is going to do is it's going to take your good old friend ATP. I'm going to use multiple here for reasons you'll understand later. And it is going to break them apart into ADP and phosphorus. And the phosphorus is going to connect right here, right? Because you need that energy. And, well, what do you do here? Well, now that the phosphorus is there, what it'll do is it'll send a signal, and the signal will then trigger three processes at once instead of just one. That's the purpose of a tyrosine kinase. It is to trigger multiple processes at the same time. Does that make sense? This one isn't that hard.
Yes.
Yep. Okay, cool. So now we have good old membranes back. And this time we are looking at ion channel. Oops. In ion channel, we have our protein. We have our ligand. There will be a triangle this time. And when the ligand connects to the protein receives a signal, and the signal causes it to open a channel. Only this time, the channel is not like the G protein channel. It is for ions. Ions being something like, I don't know, salt. Or sodium in this case. And in this case, what it'll do is it'll let the salt travel into the cell membrane, and that's about it. Ion channel is literally just specific to ions.
There's no special process to open this channel?
Oh, the ligand connected to the protein.
No, that's it?
Yeah, that's it.
Okay.
It just connects and then and then the salt gets to go through. I'm waving my hands like y'all can see me. All right, last one. There is one more receptor. It is the intracellular receptor. What is the intracellular receptor? Well, you don't need a picture for this because break down the word intracellular. It is inside the cell. So the unique thing about intracellular receptors is, well, they're the ones who can receive messages from ligands which can pass through the cell membrane. Well, what ligands can pass through the cell membrane?
Autocrine? Or no?
Not quite. Remember, polar substances cannot pass through the cell membrane. Non-polar
non-polar?
substances can. Hydrophobic chemical messengers can.
Is it hormone?
Sorry, what?
Is it hormones?
Well, no, hormones can be anything, but it is specifically hydrophobic ones. Right? These are the things that can pass through the cell membrane. And if they can pass through the cell membrane, what's the point of having a receptor on the outside, right? You're not going to be able to receive the signal. So you have instead the receptors on the inside where they can receive the signal. And that's, if you want an example, because this one actually has an example. All of your steroid hormones, right? Hormones in the shape of steroids can pass through the cell membrane. All right, that's it for chapter 11. Move on to chapter 12, the cell cycle. Now, in the cell cycle, what do we know about the cell cycle? Well, it's a big old, let me just draw that. I don't know why I put this circle in the middle, I just do. The cell cycle is divided into 4 parts. I have done this poorly, hang on. It is divided into G1, S, G2, and mitosis, which I'm going to label with an M. Now, mitosis has its own processes, but G1, S, and G2 belong to the interphase.
Yeah.
This is where the cell spends most of its lifetime, where after the cell is born via mitosis, I say born via mitosis, it grows and grows and grows until it says, hey, I need to replicate my DNA and then begin mitosis, right? G1 is your growing phase and also the phase where you are getting ready to perform mitosis. So let's write that off to the side. And preparing to synthesize DNA. Those are the big things you need to know about G1. The cell grows and develops, and it gets ready to duplicate its DNA. The S phase is where it duplicates its DNA. In the G2 phase, it checks the DNA, right? Because you can't just do mitosis immediately. You got to get ready. You got to get your centrioles all up and running, right? So here, let me do this in green. So it is preparing for mitosis. So those are your 3 phases of interphase. Pretty simple, right? All right, guys.
Yep.
Yeah, okay, good. So let's move on to mitosis. Mitosis is, well, when your cell is splitting, right? And mitosis has 5 parts to it. It has prophase, metaphase, anaphase, telophase, and cytokinesis, which ruined the chain, and it makes me sad. So, give me 1 second while I find my model for this. Okay, prophase. When prophase happens, chromosomes are clearly seen for the first time, right? Chromosomes form after your DNA is duplicated. And usually your DNA is in the form of a chromatin, right? Let's talk about chromatin versus chromosomes. Chromatin is when all of your DNA is essentially a giant spaghetti noodle. It is in your nucleus like that. It's just bundled up all together. Your DNA is not always in the form of a chromosome, right? It tends to stay like that. This, when DNA is like this, it's actually made up of two things. It's made up of DNA and histines. Let me spell that out for you. Histines, well, they're proteins. That's it. They're just proteins. So a histone, it'll be shaped like a circle, and you'll have your DNA in strands wrapped around it. Hey, that's not how you do a 3D drawing, Marcus. It looks like that. So the histines provide a bit of structure to your DNA when it is in the chromatin form. That is chromatin. So when chromatin decides to begin replicating itself, it forms itself into chromatids. Chromatids look a little something like this. One little 8, right? And this is a chromatid. It is, well, 1/2 of a chromosome, as you will remember, right? And chromosomes are literally just 2 of them, right? Because after your DNA replicates, it'll copy a chromatid into another exact copy of that chromatid. So now you have 2 chromatids. There's a bit of a binding thing there. And so what we call the 2 ends, we call them sister. Oh, dang it. Chromatids. Because there's 2 of them and they're the exact same, right? Anybody confused here?
No.
No? Okay, give me 1 second. I gotta go mute a Discord server. So, we're not done with chromosomes yet, right? So with a chromosome, what you have is you have your so you have your 2 sister chromatids here and here, but they are bound by a centromere. And it's just a thing in the middle that binds the 2 together and is used by the centriole, which we talked about a couple of chapters ago, during mitosis when they split them. And then at the bottom, you have a thing here called a telomere. They're like little caps on the ends of your chromatids. They're on all of them, by the way. Not just one, they're on all of them. But that's how it works. So now that we know chromosomes, let's go back to prophase. In prophase, the chromosomes are clearly defined, and you can kind of tell that the cell is getting ready to do its mitosis phase because, well, the nucleus is going to start dissolving, and you're just going to see chromosomes kind of like everywhere. Now, that's terrible. I'm just going to draw them like this from now on. Right? You got chromosomes floating in the soup. And the nucleus.
This is metaphase?
No, this is prophase.
This is the. Oh.
Yeah, we never finished prophase. So sometimes you'll still see part of the nucleus. They'll still be kind of contained. It'll be like kind of, it won't look very clear. But you'll see all of the chromosomes, right? Now, the next step is metaphase. While prophase is getting ready, metaphase is getting all of your chromosomes in the middle, right? So remember, metaphase, middle, both start with an M. It's lining up all your chromosomes so that your centrioles can get ready to split them. I have the chains connecting to the centrioles out here. Like that. And so what'll happen is this is metaphase. All the chromosomes are lined up in the middle. Then there's anaphase, which is literally just them splitting. So in anaphase, I'm going to redraw everything here. In anaphase, we will see there is only one on this side, separated forever. Or maybe I should start doing that. But as you can see, they are now separated. And there is nothing left in the middle. Does metaphase and anaphase make sense?
Yes.
Yes. So after anaphase is telophase. Telophase is when you have all your DNA. I'm just going to draw it as this. And it is on both ends, right? And your extracellular membrane, or sorry, your cell membrane begins to pinch like that. Oh, what did I just do there? Right? You can see it's beginning to separate. It's beginning to come apart. And cytokinesis, the final step, is when they fully break apart. That's the difference. Does that make sense?
Yes, it's pretty simple when you explain it like that.
Yeah. I like to use pictures. Pictures make the brain process better. So now, that's the whole process of mitosis, right? That's how cells begin to replicate. And I'm pretty sure y'all understand cancer cells pretty easily, right? They're just cells that refuse to die when you tell them to die. And then
They don't do apoptosis.
Yeah, they don't do apoptosis. And you're just like, oh, well, you selfish bastard. But, so yeah, in order to prevent cancer, though, there need to be checkpoints, right? There's 3 checkpoints, and each checkpoint is performed by P53. What is P53? P53 is essentially TSA, if you've ever ridden on an airplane before. It just checks the DNA to ensure that no mutations are going to happen. Kind of like how TSA checks to make sure you're, not bringing any liquids over 3 ounces. Or is it 8? Someone who's been on a plane, I don't know. So P53 is going to perform checkpoints at 3 distinct locations during the cell cycle. Let's redraw our cell cycle then. So this is G1. Yes. G2. I don't know why I made G1 so small. Hang on. And M. It is going to perform 3 checkpoints. The first checkpoint is in G1. In G1, what it is checking for is to make sure there are no mutations in the original DNA. That is the G1 checkpoint, right? Because no DNA has been made yet, right? So it needs to check all the DNA to make sure there's no damage and that nothing needs to happen before we can go and begin synthesizing DNA. If it sees damage, what it's going to do is it's going to signal other proteins, and it's going to tell them, hey, I need you to fix this cell. If you can't fix this cell, tell it to shoot itself. And the cell will commit apoptosis and die, right? Because you don't want cancer cells. So the second checkpoint, everybody understand the first checkpoint?
Yes.
All right, the second checkpoint is, well, all my DNA has been doubled, right? I have the original copy, and then I have the new copy. What it's going to check is the new copy. That's a T and an I. Ignore the fact that they're connected. So in the G2 phase, I have my original copy, which I've already checked and I know is perfectly fine. And then my fresh new copy. Well, what I'm going to check is all of that new DNA because, well, the DNA synthesizing or DNA synthesis protein could have done a bad job, and it could have accidentally, spit out some new code that I don't want and is going to cause bugs. And I'm going to be like, no. And P53 is going to scan all that new DNA, and it's going to be like, okay, either fix it or tell this cell to die. Everybody understand that part?
Yes.
All right. Third checkpoint. The third checkpoint actually happens in metaphase. Someone remind me, what is metaphase?
When the cells line up.
Not the cells.
I mean the chromatids.
No.
The sister chromosomes. Oh my god.
Somebody who isn't Alex.
The chromosomes start going towards the middle, don't they?
Yes, that is that whole part. When all the chromosomes line up in the middle. So why does it want to check metaphase? Well, if your chromosomes aren't perfectly lined up, you may accidentally pull an extra chromatid to one side. And if that happens, suddenly one cell doesn't have enough DNA, the other one has extra. Bad things are going to happen. So it checks metaphase to ensure that all chromosomes are lined up. And if they are not lined up perfectly, it'll tell cells to correct it. Or it'll tell proteins to correct it. Everybody understand the 3 checkpoints. Note, there are no checkpoints in S because S is where all the DNA synthesis is happening.
Yes.
Cool. After that, let me flip through my notes real quick. I think I am just about done. That's all of chapter 12. But there's one more thing we should go over, and that is graphs, because God knows y'all love graphs. So the main graph you will see is something that looks a little something like this. My x-axis is time, and DNA is my y-axis. And this is the starting amount of DNA. This, and then I do this, and then I do this. Sorry, they should be even. Ah. Oh, whoops. Then I do this, and then I do this. Position 1, position 2, position 3, and position 4. What is happening at position 1? G1, SG2, or mitosis?
G1?
Yes, it is G1. Why?
Because it isn't replicating DNA.
Good. So what's happening at phase 2?
It'd be the S phase
Why?
because it's replicating DNA.
Good. The reason why it's not the G2 phase is because at G2, when G2 starts, you have double the amount of DNA, which makes phase 3 G2, because now I have double the DNA. So what's phase 4?
The cell when it replicates, like when it's already done with mitosis.
Right, but what phase is that?
There's a phase for that? Was it cytokinesis or no?
No.
No, I don't know.
Anybody? Come on, guys. I need participation. Please, it makes me feel better. Ah, damn, all right. It's G1. Remember, it's a cell cycle. Right? Because mitosis actually happens here, right? When it's splitting. And once it's fully split, it just goes back into its G1 phase. That's it. Also, we didn't talk about G naught or G0. When during the cell cycle, redrawing this bad boy. G1, S, G2. During the cell cycle, you have G naught, which is essentially when certain cells will do this. When they enter G naught, they just don't replicate their DNA. They perform all their functions. They just don't replicate their DNA and they don't split. The main cells that do this are your nerve cells, which is why drinking alcohol is bad for you, because it kills your nerve cells and your nerve cells don't replicate. So, I mean, you have a couple million to spare, but you know, that's a lot of damage. No, I'm not about to do that. Anyway, that's what G naught is. Everybody understand that?
Yes.
All right, that's it. We're done. Any other questions y'all have for me?
One more time, could you explain the chromatin, chromatid?
Okay, chromatin versus chromatids versus chromosome. Okay, just know their shapes.
Oh,
Sorry, what was that, Katie?
Accidental, my bad, Marcus.
Okay, so the difference between a chromatid, and a chromosome. Chromatin doesn't have a shape. It's just a giant bowl of soup. It's all this stringy mass of DNA. A chromatid has this shape. It is one half of a chromosome. A chromosome is after you duplicate the chromatid, and it looks like this. This is a chromosome. Does that make sense? That's the difference. Just know their shapes.
Okay, I got it. Thank you.
Okay. Anybody else? Is there anything else I have missed? No, looks like that's it. Oh, I didn't cover this. Synapses. Imagine, for instance, you have a nerve cell. It's got the weird scientist hair end. Got a big tunnel. You got this bit. So imagine that's a neuron, right? A synapse, in this case, will send an electrical signal. Sorry, I should do it in yellow. It'll send an electrical signal rather than a physical signal to the end, telling the neuron to send a specific ligand to certain cells. So that electrical signal will travel through that. It'll send out specific synapses, and they will make contact with whatever is on this end. And that end has its own receptors. Ready to cling on to those freshly made ligands and do whatever response. And usually that response is feeding me dopamine because I just finished a review. Everybody understand that? That shouldn't be too hard. If you see this, just know it's a synapse.
Yes, I got it.
All right. Remember, the word hormone and ligand can sometimes be used interchangeably. Remember that a hormone made in one place. Oh, whoops. Used in another. Y'all got that?
Yes.
All right, cool. That's chapters 11 and 12. And that's everything I missed from my notes as well. Any other questions before I let y'all go and pray that y'all pass this test?
Wait, I heard that you know how like we're supposed to take like a quiz for the whatchamacallit. You know how we after the project we have to take a quiz? Is that gonna be tomorrow too or?
Yes, that's going to cover chapter 3. But just look at my videos from cellular respiration and photosynthesis for that.
All right, bet.
For the cellular respiration one, skip to like halfway through. Because that also has chapter 8 in it at the beginning half. And you don't need that information. Or, well, you won't need most of it. So, that's it. Watch my other videos if you want help on that test. I'm going to say bye to all of you. Good luck on your test.
Thank you, Marcus.
Remember, who's in here? Katie, Alex, Clark. Who else? Luis, Mia, Jessica, KG, and Simon. If all of you pass, maid outfits getting whipped out on Saturday, all right? Well, not Saturday. Some Saturday. All right, all 9 of you have to pass, though.
So just, wait, you counted yourself in 9, didn't you? Or is that not how
Oh,
this
sorry,
works?
no, that's 8. That's 8. I'm not counting myself. I'm allowed to fail. I get special rights being the guy who's doing the review. Anyway, that's it. All of you go pass your test, make me wear a maid outfit. I got really disappointed last time because somebody failed. I swear to God, I'm not gay. But anyway, I am done.
Somebody told me.
Marcus, can I talk to you after this?
Yeah, sure. Let me end the recording.